Hello, everyone. This is Dr. Shiva Ayadure. Most of you know I'm a system scientist. I've been doing this for a long time. CytoSolve, a technology I created, is really a powerful engine to understand at the molecular level how things work based on the known science and aggregating science. So we're going to talk today about how mixing supplements can have positive or negative effects. Many of you know I'm a big proponent of science. I'm also a big proponent of figuring out what are the right supplements for the right person. Food is medicine. And so we have to, even whether it's supplements or food, we have to be conscious about how we put them together because each one of our bodies is different. Now, the technology I created out of my PhD work allows us to get a handle on that, taking it away from being just a black art. So we're going to talk about, this is the first of a series I may do, and we're going to look at two compounds, arginine and caffeine. Arginine, as you know, releases a lot of blood flow, and caffeine, stimulates our body in various ways. And how do they mix together? Should you take them? Should you not? So today, what I want to talk about is applying system science, molecular system science, to really understanding the synergy of supplements or the city. And the good news is just about a couple of months ago, I was a co-author, one of the principal authors on a major paper that came out on dietary supplements. It took us 5 years to get this published. That's how these publications take place. I'm pleased to share that with you, but I want to teach you that when you take multiple things, the average 82-year-old is on 12 different drugs. The average 82-year-old. No one even knows how they interact. Well, CytoSolve allows us to understand those interactions long before we kill animals, etc. So if you're ready, let's begin. Some people call it a polymath. In addition to creating the first email system, but I also run various companies. One of the companies I run, I do it because I enjoy doing it. If we make money, great. If we don't, that's also fine. But CytoSolve is an amazing technology that came out of my PhD work, which really allows us to model using the computer different biological functions. We can model Alzheimer's, we can model cancer, we can model different functions like inflammation, etc. on the computer. This allows us to understand toxicities. This allows us to understand synergies way before we go kill animals, way before we even waste time in the lab. I wanted to let you know that we use the technology here. We've helped many companies over the last 16 years, a lot of smart, innovative companies, but we decided with all the mathematical models we've created, why don't we try to use this to compute the best product we could think of from the science out there for reducing pain and inflammation, pain and discomfort. And that resulted in us creating MV25 using CytoSolve. We're going to have more products that are going to be coming, but let me just show you what MV25 is about for those of you who haven't heard about it. But this is using CytoSolve in a beneficial way, not to just do research, but find combination therapies.
Hi, I'm Barbara Ann. My hands would cramp up so that I couldn't hold cards or knit or crochet, and they would go like that. And I'd have to use this when I played cards with my grandkids. And I started taking that MV25. After a bit, I was able to hold cards in my hand, very little cramping, hardly at all anymore. MV25.
Hi, my name is Sandy. I'm a Taekwondo instructor. I tore my ACL during Taekwondo. I had a lot of pain and limited mobility. I've been taking the MV25 for about 6 months now. After the first week, I noticed a big difference. After the second week, almost literally no pain.
My name is Jeremy, and I suffer from a lower back problem. Hurt my back at work years ago, and I can go to the chiropractor, do all kinds of different things, and nothing seems to help. And I decided to try MV25. I didn't notice a difference immediately, but within a few days, the pain went away, and it stayed away. I've continued to take it, and even when I do things that I shouldn't do, it seems to go away a lot quicker than it ever did before.
It's clean food certified. It's made in the US. If you go to vashiva.com, right on the shop, you'll click there, or you can go right to mv25.life, either way. And then from there, you can click on the bottle and you can order. If you buy 6 bottles, you get 6 bottles for free. Please take advantage of it because first of all, it's going to help you. It's going to help our movement, and it really supports the fact that we want to take science-based approaches to natural products. As I've shared with you before, the way that pharmaceutical companies, which are considered, by the way, the gold standard of all the different testing they have to do, it takes them around $5 billion in 13 years from the time they discover a single ingredient. Let's say something like Lipitor, which they use for heart disease. As they find it in the lab, it takes them, first they have to do lab testing, then they do kill animals, then they go through human trials, phase 1, phase 2, phase 3. That takes around 13 years. And at the end of it, by the way, that drug that they find, it only works for 10% of the people. No one tells you about this. The other people have all sorts of toxicities. Now, 20% of it only makes it through phase 1. So imagine if we could figure out the toxicities and efficacies way ahead, how many lives we could save, how much money we could save. That's what really led me to the development of CytoSolve. Now, those of you need to also understand pharmaceutical companies are actually burning to the ground. That bar chart right there, the blue bar chart shows you how much money they keep spending. And you know what? They're finding less and less new drugs. Not only finding them, the FDA doesn't even approve them because even the FDA is saying that they can kill people. So that's that orange line. They spend 30% year over year in R&D, and they're actually getting less and less new drugs out the door. This is the truth. My feeling was the reason this occurs is medicine doesn't treat the body as a whole. They treat it like a bunch of parts. One guy's working on this and another guy's working on this. But if we could put it together as a whole, that's a systems approach to medicine, we could solve a lot of things. Now, the good news is in 2003, even MIT decided to take a systems approach to medicine because what had occurred was, as I've shared with you before, in 2003, the Genome Project ended. And when the Genome Project ended, we found out we only have 20,000 genes. We don't have 1 million genes. We don't have 100,000 genes. We thought that we were more complex than a worm because of the number of genes. It's not true. It turns out we have the same number of genes as a worm. So that led to the field called systems biology. And one of the cool things, the reason I came back to MIT at the age of 40, running a very successful company, is because I always wanted to use computers in medicine. And one of the ideas that was coming was, hey, we need to take a systems approach to medicine. If we want to understand the whole body on the right here, we need to interconnect all the parts. And one of the challenges that the National Science Foundation put forward was, could you mathematically model the whole human cell? And by the way, these are individual molecular pathways in the cell. And if you could do these maps and interconnect those models, you could get a realistic understanding of the human cell. That's why I developed CytoSolve. CytoSolve allowed us to do this in a very powerful way by interconnecting parts while computing the parts separately so you could mathematically do this. Otherwise, it would have been impossible. So that was CytoSolve, published a number of papers on it. And by the way, this is how Big Pharma does. What I realized, what I discovered with CytoSolve, you notice we can handle compounds, combinations, we can handle food. So on the computer, long before we even do testing on human cells, we could do it on the computer. And by the way, this is how we build airplanes. We take a design, we don't just throw a test pilot and we don't go to wind tunnel testing, we test it on the computer. We figure out all the mistakes. And that's what CytoSolve allows us to do. And we've created a very powerful engine to do this. And we have the ability, so we don't just cherry pick. That's what the establishment, that's what the deep state does. They like to cherry pick. We take all the information and we take all of that's in the literature, we extract it, we digest it with CytoSolve, we figured out the molecular pathways, and then we interconnect them to really give a realistic understanding. So today, we're going to talk about the toxicity of caffeine and arginine on cardiovascular function. A number of you, by the way, if you want me to do different kinds of tests, different kinds of videos, I've gotten great ideas. Believe it or not, we actually take your ideas. We've created a massive blackboard, and we're going to start going after each one. But a number of you said, hey, we'd like to understand cardiovascular function, cardiovascular disease. The number one source of death in the United States is cardiovascular disease, heart disease. In India and China, it's liver disease. What I want to start with you is really looking at cardiovascular function. And by the way, to those of you asked, this is a paper that I just published. It's called Multi-Decision Criteria Analysis Model for Assessing Risk from Multi-Ingredient Dietary Supplement. This was just published in the Journal of Dietary Supplements, a very good journal. You can pick it up. It's available online. And if you notice, I'm one of the senior authors on it right here, Dr.SHIVA. Very proud of it. So I want to point that out. It took us 5 years to get this published. But what I want to talk about, first of all, is cardiovascular function. Now, one of the most important things that I just want to step back and talk to you about is when your heart is functioning, right? When you're actually using your body and you're doing exercise. One of the important things, the reason exercise is important is your body pumps blood through your arteries. And when that blood is pumped, guess what happens? When you exercise, your body sends blood through all the infinite number of, not infinite, but all your arteries throughout your body. And when that blood flows through your arteries, you know what happens? An important molecule gets created called nitric oxide. Nitric oxide. I believe Time magazine, one of the big magazines, called it the molecule of the decade. The reason nitric oxide is important is an important source of health. When you exercise, your body releases nitric oxide. In fact, nitric oxide was a basis for creating Viagra. Viagra is when people have erectile dysfunction, they don't have enough blood flow, they don't have enough vasodilation. So nitric oxide is central to that, and that's what that chemical does. Viagra, it creates nitric oxide release. But you don't have, that's not the only way. Exercise does it. There's many other nutrients we'll talk about. So let's go back to the slide set here. So when you look at it over here, what I want to share with you is several years ago, when I was at MIT in my PhD work with my collaborators at MIT, we actually were looking at how nitric oxide is released, how it's metabolized in the body. study. This was a work between my lab at MIT, Harvard, Brigham and Women's, and King's College in London. Great, amazing people we work with, really great researchers, not the institutions, but I'm talking about these scientists, good people. So when nitric oxide, what you're looking at here is, this is a piece of the artery. You're looking at a cross-sectional view of the artery. And what you're seeing is when blood flows through your artery, right here, nitric oxide gets released right here in the presence of something important called L-arginine. Nitric oxide gets released in the presence of arginine when blood flows. So blood flows through your arteries. In the presence of arginine, nitric oxide gets released. Now, how does that happen? Now, Andrew Koo in our lab, great researcher, he had meticulously set up the wet lab experiment where he could send flow and he could measure NO release. But he wanted to figure out how this was occurring. Now, if you look at the surface, of the artery, in many ways, the surface of the arteries are like bathroom tiles. If you ever go into your bathroom, all those tiles that you have, well, the surface of the artery is composed of similar tiles called endothelial cells, to be specific. And that's what the zoom I'm doing here. These endothelial cells here have a little Christmas tree structure, if you see right here, called a glycocalyx. And when blood flows over that Christmas tree, which is on the surface of the endothelial, it moves. It's called a mechanotransducer. The motion converts itself to mechanical function, and guess what? Voila, nitric oxide gets released. Now, how does this happen? Well, it was very hard to understand. That's where CytoSolve came to be very powerful. And what we found was, let me show you right here, if you look in the literature, it's like the blind man following the elephant. Everyone has their view of nitric oxide release. This is why we don't get a truth. Everyone has the pieces, but with CytoSolve, we put them together. Some people believe it's calcium influx. Other people believe it's phosphorylation. But with CytoSolve, we're able to meld all of them together without bias, objectively. And we're able to do here is put all these little chemical reactions together with CytoSolve and then mathematically model it. And what we see right here is this line here. Behind this line is all those integrated mathematical reactions, which could not be done without something like CytoSolve. And that line predicts how much nitric oxide, or the protein that creates, or the mRNA that creates nitric oxide, will be released. So it's a direct relationship to nitric oxide. So that's our prediction. Now watch this. It's quite impressive. That orange dots are the actual wet lab experiments from the laboratory. So what we're showing is that is a prediction, which is a black line, and the orange line is a wet lab prediction. So I want to just take a step back. This is why CytoSolve is so powerful. Our predictions actually match what people were seeing in the laboratory setting. So this has never been done before. Until we did this, no one believed that you could use the computer to do this kind of modeling. And same here, we're looking at the actual eNOS protein, and same thing here. Bottom line, behind that black line is all those chemical reactions, and we're able to predict it with CytoSolve. So, by the way, this was published in one of the most eminent scientific journals in the world. We got music here, by the way. You don't get science and music in any other place but right here on Dr. Shiva's Live. But what we published this, and it was an important publication because, remember, these guys are the establishment scientists. They don't like to publish something unless they think it's all in alignment. Well, we got it published in one of the journals, and it shows that even they blessed our work. It's very important because they can't now say CytoSolve's garbage. They, in fact, validated it. So this was published in Cell. So bottom line is, we had this very powerful model, which had been published in one of the most eminent journals in the world, showing that CytoSolve could be used to predict cardiovascular function. What do I mean by that? What I mean by that is the release of nitric oxide is central to cardiovascular. You have good nitric oxide release, that means your heart is working well. That means your cardiovascular system's working well. Is that clear? So we had done this work. Well, several years after I did this work, I was invited to give a talk at Walter Reed Medical Hospital to a room full of 200 doctors in the military, in the Army. In fact, the Army Surgeon General was in the Army. And when I finished my talk, they were very impressed with what the technology I created. And they said, hey, Dr. Shiva, we'd like you to be an expert on the U.S. Pharmacopoeia. U.S. Pharmacopoeia is an organization which tests supplements. If you go to Whole Foods, you go to your health food store, you'll see a call UE. They're the ones who validate are the ingredient levels in the supplements or supplement manufacturers are saying. So I said, sure. And I wanted to help them out. And the reason the military was interested in this, soldiers, our veterans, take all different kinds of supplements to support their, for example, to be strong, muscle strength, to be alert. They take supplements like arginine and yohimbe and caffeine and creatine and synephrine, bitter orange. So soldiers were taking these 5 supplements. Individually, they may be good, but what do they do in combination? And they didn't know how they actually worked. So they'd set up this expert council to figure out their working. So when I joined the expert council, I looked at the way they were doing, and they were doing it in a very, frankly, a dumb way, using spreadsheets. They would ask soldiers if they came in with a headache, hey, what supplement did you take? And they would note it down on a spreadsheet, and they would try to correlate that observation to which supplements are taken. It's called statistics. And I said, look, there's a smarter way to do this. I recused myself and I said, look, I will independently offer you CytoSolve as a public service. And we took 2 supplements, arginine and caffeine and synergy. And we wanted to show them the power of CytoSolve to understand synergies and toxicity. So that's the results I'm going to share with you right now. It's really cool. And again, you can see the logo of Walter Reed Medical Hospital. Here's USP, US Pharmacopoeia. So we were commissioned, and we did this as a public service to the government, to look at caffeine and arginine together. So what we did was we took all of those nitric oxide pathways and we looked at arginine and caffeine together. Remember, we'd already modeled this. And we looked at people who had different ADMA levels, which means hypertension. And let me explain this. So what we did was, this was our first experiment we ran on the computer. No animal killing, all done on the computer. And what you're seeing is 4 sets of bar charts at different levels of arginine. This is a predictions of CytoSolve. No animals were killed in this experiment. But here what we see is we gave different levels of arginine, 40 units, 80 units, 116, and 300 units at the cell surface. Now, these different bar colors are people with different levels of hypertension. Blue means you're normal, you don't have hypertension. Okay, which means you have low ADMA, high ADMA, which is denoted by the purple, and it means you have hypertension. What do you see here? Well, let's look at it step by step. What CytoSolve predicts is, as you increase more arginine supplementation, guess what happens? You get more nitric oxide release, okay? So if you're at 100%, which is, if you look at the blue, which is most of us, if you're normal and you don't have heart disease, yes, nitric oxide helps you, get you up to about 110%. So it has reasonable effects. But if you have seriously high hypertension, which is that blue, that purple graph, and some of you, if you have hypertension, which means high blood pressure, you notice arginine really helps you a lot. It takes you from about 65% levels of nitric oxide release all the way to nearly 90+%. So arginine is very powerful to release, to support vasodilation. Then what we did with CytoSolve, so we did that independently. And by the way, this matches all the clinical data people have seen. Then we said, what happens when you do caffeine? By the way, there's supplements you can get at the health food stores. You take 1 shot, it's like taking 40 cups of coffee. Some people take it if they want to stay awake, hang out, party. Soldiers sometimes are taking this to stay alert. Well, you notice what caffeine does. Our research shows, as you take, this is 1 cup of coffee, which reduces nitric oxide from about 100% to 85, 90%. But if you take 40 cups of coffee, some of these supplements do, or 10 cups of coffee, I think it's 10 cups, I'm sorry, you bring down your nitric oxide levels down to 60%. So we were able to show that, and people have seen this clinically, but we were able to show this on the computer and match it. Now the question is, what happens when you combine it? So let's ask this question. What happens if you have a soldier who's taking arginine supplementation and he's taking these shots of caffeine? I think those high energy things have caffeine in them, right? So what happens if you're taking arginine, you're bodybuilding, you want to get the pump, right? And you're taking caffeine. What will it do? What are the synergistic effects? So on. So what we're doing here with CytoSolve, we asked that question. So what I'm going to share with you right here, make this graph a little bit bigger so more people can see it. Hope that's better. What I'm going to show you, the left graph and the right graph are different. The left graph is I'm taking arginine with 1 cup of coffee, regular 1 cup of coffee. The right graph is when I'm taking arginine with the supplement called C4, which is like taking 10 to 40 cups of coffee. What you see here on the left side is, as you take coffee, guess what happens? Your nitric oxide levels goes down. You see, this is very different than here. You're at 100% in the blue, but look what happens. If you have normal health, look at the blue chart on the left side, you're at 80%. And guess what? Arginine helps you get back up to 90%. So it's good for you. If you're taking lots of coffee, nitric arginine can help you get back to normal levels. But look what happens to these people who are hypertensive, high blood pressure. Notice the 1 cup of coffee already increases their blood pressure, which means makes them more hypertensive, down to 50%. And look what happens. Arginine does help you, and it helps you get back up to sort of normal, about 70, 80, close to 80%. Okay, so you could argue you take 1 cup of coffee, you take arginine, it helps bring you back to normal. Now look at the right graph. You're taking 10 to 40 cups of coffee with those supplements. It seriously depresses you down to around 65%, even for the healthy person. For the hypertensive person, the purple graph, close to 40%. You really increase your blood pressure, you increase hypertension. And guess what? No matter how much arginine you take, it only gets you up to 60%. So what our research was able to predict and tell the military is, look, if you have soldiers who have high blood pressure and they're taking arginine, they think it's going to help them, and they're taking that high dose of C4, caffeine, 10 to 40 cups of coffee, it's not going to bring them up. So they should not be doing coffee, no matter how much arginine that they're doing. Does this help you understand? So we're able to use CytoSolve in these very powerful ways. And look, I am going to always tell you the truth. CytoSolve is a scientific source of truth. So we got to remember, supplements, food, these are all medicines. Some people say, oh, I'm going to eat, X amount of turmeric. I'm going to eat so much arginine. Well, all of them have limiting factors, as you can notice in this graph. No matter how much arginine you take, at a certain point, it's called the arginine paradox, the blue graph here, the blue bars. It doesn't help you. Some people think more is good, and it's not true. So here's a very powerful way in how we can understand how to mix or not to mix. So in this case, what we're saying is, if people have high hypertension, be very careful in taking high doses of coffee and thinking arginine is going to bring you back up. And by the way, what I really am very proud of was the CytoSolve technology became published in the monograph of the USP, which is US Pharmacopoeia, which is a very prestigious organization which tests these supplements to validate. It's a gold standard. So CytoSolve was the core of that, and I was proud to be one of the co-authors in this publication. But in summary, what we've shown here is that we can get quantitative, predictive insights into toxicity levels of caffeine and arginine. We can also predict the nitric oxide levels tailored according to physiology and pathophysiology based on different levels of high blood pressure, low blood pressure, and all the states in between. So CytoSolve emerged as the only platform that could analyze toxicity effects. Now, thank you, everyone. I got to go to a call, but I hope you enjoyed this. Be well.